Hello everyone, welcome to Aakash. This is Lavina Singh, your botany mentor, and in this session I'm going to discuss the slot 2 questions with you, and slot 2 comprises of these chapters, that is biological classification, anatomy of flowering plants, plant growth and development, molecular basis of inheritance, and biodiversity and conservation. So this is the discussion of AIATS Plus 2025 phase 2. Your test number was MT07, that was conducted on 2nd of March 2025. I hope you people are preparing well for your exams, and exam is pretty much overhead. Your board exams are also going on, a lot of pressure, right? But no need to worry, just thoroughly revise your concepts. Please take care of your health, and also please pay attention to your NCERTs very well, okay? You need not read beyond NCERT, you can simply dig into your NCERT, concentrate on all the particular topics well, sharpen your concepts at the last moment, and you will be perfectly giving the best results to your parents, right? So in this particular session I have already told you the chapters, and since it is slot 2, I will be discussing the questions that are selected in which you make frequent errors from these very chapters, okay? So the first question that we are going to discuss today is question number 102 from test MT07, which says during aminoacylation of tRNA, okay? So aminoacylation, when does it occur, why does it occur, right? We need to understand the concept first. You know, these sessions that we are conducting on YouTube, they are live for all of you, and very important thing about these sessions is it is not only the discussion of the answers of these very questions, but also the related concepts, okay? So the concept behind this very topic or this very question is translation, right? So while we are talking about, this is from your molecular basis of inheritance, right? So the topic is translation. What do we understand by the process of translation, right? What does exactly happen in this particular process? Basically, while we are talking about the polymerization of amino acids, okay, basically protein formation is taking place from RNA to proteins, that is translation. We know about the particular process, the chronology, replication, transcription, translation, right? So central dogma says that. So translation stands for polymerization, okay? If you understand each term from this very definition, you will understand that what is happening in this process and what is required for the same, okay? So polymerization of amino acids, right? Now what do we understand by polymerization? That means formation of a polymer. Polymer means a whole chain, right? So polymerization of amino acids to form a polypeptide, okay, to form a polypeptide. Now from this very statement, please pay attention, what does it say? It says that formation of polypeptide is taking place. What do we understand by that? See, polymerization of amino acids, matlab a long chain of amino acids. So we have one amino acid linked to the other, then to the other, and so on, right? So this is one amino acid, second amino acid, and so on. It is forming a whole chain. Hello Pragati, how are you? Now here we have the presence of these bonds, right? And these bonds are nothing but the peptide bonds. These are the peptide bonds, right? Now the formation of peptide bond requires energy, okay? And we need to know from where does this energy come, okay? So we have the formation of peptide bond that is taking place over here. We require energy for the same. Now coming back to the process of translation now. Translation is saying that polymerization of amino acid is taking place. Now the first step of this particular process is the presence of tRNA, and this tRNA is getting charged up, okay? What do we understand by the charging of tRNA? Here I am drawing a rough representation of tRNA. It is having its 3 loops, right? And over here we see that this is the 5' end and this is the 3' end, right? This is what we learn. Now this 3' end represents the amino acid acceptor end, right? So this end, this 3' end is basically amino acid acceptor end. That means whichever amino acid is present will come and bind to this position, okay? And let us say this is the amino acid, it has come and bound to this particular position, okay? Now what will happen next? Now this particular process that is happening, that is binding of amino acid to the 3' end of tRNA, is happening in the presence of energy, that is ATP, okay? So it is requiring the presence of ATP, right? It occurs. Now what happens is this particular charged tRNA, what it has become? It has got the amino acid on its 3' end, it has become a charged tRNA, okay? And this is the same that we call the aminoacylation of tRNA, okay? Very good morning all of you. Now we have aminoacylation of what? Of this tRNA, okay? So aminoacylation of tRNA, that means our transfer RNA. What do we understand by this? How do we define aminoacylation of tRNA? It means that binding of amino acid, that is why it is aminoacylation, right? Amino acid to its cognate tRNA. Okay, now what do we understand by cognate tRNA? Okay, see, for example, this amino acid that has been represented over here, let us consider it as tryptophan. Okay, so if this particular amino acid is tryptophan, okay, and it will have, what is the codon for tryptophan? If I am considering mRNA, okay, so I'll make it over here for you. Here is the mRNA, right, to which this particular tRNA will come and bind. Okay, so this is the 5' end of mRNA, this is the 3' end, and here will be some code, right? That code will be particular for tryptophan, right? And for that the code is UAC, right? Now to bind to UAC, what is required? An anticodon, okay? So this cognate tRNA, what do we understand by cognate tRNA? That means for this amino acid tryptophan, it should have an anticodon on this particular loop, okay? So this is called as the anticodon loop, and that is why it will be having the codon AUG, right? So it is complementary to this, right? So this particular tryptophan will be binding to its cognate tRNA, and this is happening in the presence of the energy hero, that is ATP, right? Now this charged tRNA is bringing energy, right? Now what happens, you can see in this particular representation that over here, when the tRNA, that means the transfer RNA, will bring the amino acid. Now this is the whole setup. We have the smaller unit of RNA, we have smaller unit of ribosome, the larger unit of ribosome, it is binding to the mRNA, and the process is going on, right? Now what will happen? This tRNA is bringing amino acid with it, okay? So this tRNA over here, we can simply see in this representation, this tRNA is bringing an amino acid with it, right? It will land at the A position. We have the E, P and A sides, right? It will land at the A side. It will shift towards the P side. Okay, this is a train like fashion going on, right? It will keep on running like that. So when A will shift towards the P, what will happen? It will bind to the already present amino acid over here. So we have the presence of tryptophan already, right? And this is bringing phenylalanine over here. Now what will happen? It will shift towards P side and it will bind to the tryptophan as represented in this very representation. Okay, now you can see that this binding that I told you in the very first statement requires energy, and that energy to bind, over here we see tryptophan, it is bringing phenylalanine, and for this required energy will be in the form of this ATP, right? So what is required? Energy from charged tRNA, right? That is ATP. Along with that, there is the requirement of GTP also, as well as there is the requirement of a catalyst. Okay, now what is happening? These three factors will help in forming this peptide bond. Okay, that is why here will be the binding of one amino acid to the other, in this case phenylalanine to tryptophan. Only then a whole chain will be formed, protein will be formed, right? And this energy is coming from charged tRNA, that is this one. We have GTP as well as catalyst. What is this catalyst doing? It is basically helping to reduce the activation energy. Okay, suppose for a certain amount of for a certain work you require 100 joules of energy. Okay, but if we reduce that energy to 50 joules, the work will be done in a simpler way, in the easier way. So this is what this catalyst is doing. It is reducing the activation energy. That means the bond formation that is requiring the energy will be reduced. Okay, so the process will be facilitated by this very catalyst. So ATP, GTP and catalyst will be helping in the formation of this bond. Okay, so this was the related concept. Okay, very congratulations to Ayush Singh Rajput. There will be many more such news that the students are getting placed to the very reputed institutions if you keep on practicing and learning better. Okay, so we have, switching back to the question now, the question asked about aminoacylation. I told you about the related concept also. Now what is aminoacylation? Amino acids are linked to their cognate tRNA. Yes, that is what aminoacylation is. Second option is initiator tRNA binds to AUG codon. No, this is not true, right? Then amino acid binds to ADP, this is also not happening, and tRNA binds to the stop codon. No, this is also not true. So aminoacylation, as the name states, amino acid के साथ bind करना, right? Thank you so much, Pragati. So this is the correct answer for the given question. Now moving on to our next question, that says, out of the given mRNAs, which will produce produces protein with maximum number of amino acids. Okay, so we have been given these particular mRNAs. Okay, now we have to understand what exactly happens when the process of translation is taking place. Okay, so while we say that there is the presence of total 64 codons, right? We have learned that from NCERT also. There is a whole table given over there of total 64 codons, right? So total codons are basically 64 in number, okay, and generally 61 out of those code for amino acids, right, and rest of the 3 are called as the stop codons. Okay, so only 61 they are coding for amino acids. there is an exception also that I will be discussing in the further question. Okay, so generally 61 codons are coding for amino acids. So the remaining 3, they are not coding. That is the meaning, right? So the 3 codons are basically the stop codons. Okay, so these are the stop codons. We also call them as the nonsense codons, right? So they are also called as nonsense codons. Now we have to keep in mind that which are these 3, okay? So you need to memorize these, that the stop codons are, बाकी सब आप याद करें या ना करें, these stop codons are very much important, okay? Hello Ishta Kaushik. Okay, you are a NEET aspirant for 2026. Very nice. Very good, very good preparation is required for that. You already know, right? All the very best for your preparation. Very good morning, Keshav. Okay, so here we have the 3 codons that are the stop codons. And which are those? We have UAA, UAG and UGA, right? We have also given them names, right? This is the ombre, we have, okay, there is something that is, okay, we have ochre, amber and opal. These are the name of the gemstones, okay, that we have given to these 3 gems that are nonsense, okay. So ochre, amber and opal. Now we have to see in a particular sequence if there is the presence of any of these, the process of translation will stop, because their function is to stop the process of translation, okay. Now if they are stopping translation, some particular code has to start also, right, the process of translation. So we have one codon that codes, that is AUG, and it is the start codon that basically starts the process of translation, okay. So 3 stop codon, 1 start codon, okay. Now let us switch back to the question now. This is the sequence that we have been provided. In the first, second and third sequence, we can see the positioning of AUG over here, right? We have AUG, again we have AUG and AUG. That means start codon is present at the very first position. Now we have to see where is the stop codon, right? So we have in the first one, we have the presence of UAG over here, right? That means the process of translation will stop right here, right? In the second case, we have the presence of UGA over here. This is also a stop codon. And in the third one, we have the presence of UAA, right? And this is the stop codon over here. So start codon, stop codon. The distance you can clearly see, that is very much clear over here in the third case, that which will produce the protein with maximum number of amino acids, right? So the longest chain will be forming the maximum number of amino acid. So the third, C option is the correct one, and according to this, we have the third as the correct answer, right? Arush, is it clear with you? Good. Krish, Study Empire, everyone has given the right answer. That means we have to see the start codon, we have to see the stop codon, and then we simply see the length. Okay. So in this case, we have the longest one in this case, and that is why it will form the maximum number of amino acids. Clear with this? Now moving on to the next question. Question number 113, which says, which one of the following is not explained through the species area relationship curve? Okay, please focus on not. Okay, sometimes we are in so much hurry, we simply read the question, we are so excited. Species area relationship, ये तो बहुत अच्छे से आता है. Let's answer. और पहले ही आंसर को टिक कर आ जाते हैं फटाफट. Okay, we do not have to hurry, we have to pay attention what the question is asking. Okay, so for this, let us understand the concept first, that is of species area relationship. What does it say? This is the graph from your NCERT. And this, okay, can you tell me in the comment box who gave this curve, who gave this relationship? Please tell me in the comment box. Species area relationship curve. Okay, we will understand this particular curve. Okay, and who gave this curve? Tell me in the comment box. It was given by Alexander von Humboldt. Okay, and that is why it is also called as the Humboldt curve. Okay, now the question is asking, whatever the question is asking, there are so many characteristics that have been mentioned in the question, in the options. We have to understand the curve and then we will answer the question. Okay, now yes, very good Rohit, von Humboldt gave this curve. We have, okay, now what do we understand by this? This particular species area relationship says that as we increase the area, species diversity also increases, but up to a certain limit. Okay, that means after a certain point, you keep on increasing the area, but the species diversity will not increase further. Okay, now this particular curve, if we take up the log scale, right, so we can see that it is giving us a straight line, right? On the graph, it will give us a straight line, just it has been given in your NCERT, right? So, and if we are speaking about this is giving us a straight line. And for a variety of taxa, we will consider it as the rectangular hyperbola. Okay, please do keep this in mind and always read your NCERT properly, because the questions that are asked are very discreetly asked, right? Some statement that has been given in your NCERT, from in between the statements you will be given the question. Okay, so please do read them properly. Okay, so for a wide variety of taxa, large variety of taxa, we see that the curve is the rectangular hyperbola that has been given over here. Now the equation that is for this particular curve is log, I'm so sorry, we have log S is equal to log C plus Z log A, right? This is a very important equation and the hero of this equation is Z. Okay, we will understand what rest of these are also. First of all, if we take up S, S represents what? You can continuously answer these in the comment box also. What does S stand for? S stands for species richness, right? Right? If we are taking up C, C is, what is C standing for? Tell me in the comment box quickly. C, we have Z. Okay, first I'll write A and then I will tell you about Z. Okay, so we have A and then we have Z. Okay, C is representing y-intercept because we are taking it on the log scale. So we have the y-intercept. Okay, then we have A, that stands for the area, and at last we have Z. Z is basically the regression coefficient. What do we understand by regression coefficient? It is basically representing the slope of the line. Okay, this is the line that is showing a slant, right? And this basically the steeper the slope, the more is the value of z. Okay, what do you understand by that? See, if we are talking about an area, okay, I'll draw a graph along. Okay, if we are considering an area, on this scale we have area, on this scale we have the species richness. Okay, you will understand what is the slope. Okay, pay attention very carefully. Now we have this much area, and in this much area we have the species richness that is abundant. Okay, species richness is somewhat here. Okay, very good, very good. Everybody is answering. Now we have this particular plot, right? Now if I want to draw the slope, okay, so the slope will be like this, right? Now we need to compare it with the other scenario. Now I am increasing the area, okay? The increasing area has reached till this particular point. Now what does this particular relationship say? It says you keep on, you increase the area, species richness increases. I increased it till here, the species richness was increasing, but up to a certain limit. Now if I am increasing the area, the species richness will stay constant. Okay, so if the area is this much, now the species richness is reduced. Okay, the richness is reducing. So what will happen to the slope of this line? It will become less steeper. Okay, draw it again for you. Now this particular slope, you can clearly see this is the slope x, this is the slope y. Which one is more steep? Which one is more steep? The x1 is steeper as compared to y. Okay, so it is telling the same, that as you increase the area, species richness increases, but up to a certain limit. Okay, so the slope of the line is being represented by z. It is also called as the regression coefficient. Okay, so, or also called as the slope of regression. You can be asked in any form, right? So here we have the x that is more steep. Okay, now this is what the particular relationship is about, right? And here is the representation that has been given in your NCERT. This equation you should learn by heart, okay? And the scale you should be knowing that this is how we represent it. And if we are being given that for a wide variety of taxa, what sort of shape is formed, you can simply answer rectangular hyperbola. Okay, now coming back to the question. Which one of the following is not explained through species area relationship curve? So the first option says the slope of the line is much steeper for a very small areas. I told you just now, I told you if the area is smaller, the slope is steeper. Okay, then on the log scale, that means this is correct. Okay, on the log scale, relationship between species richness and area is a straight line. Yes, we just discussed this. Species richness increases with increasing explored area, but only up to a limit. That is also absolutely correct. And the fourth option says the relation between species richness and area is rectangular hyperbola for a wide variety of taxa. That is also correct. Then where is the... Okay. Over here, on the log scale, we have the species richness and the area is a straight line. Okay, species richness increase. The first statement that you are seeing over here, what should be the correct answer for this? It is asking that is not explained in the species area relationship curve. The slope of the line is much steeper for very small areas. Okay, so here will be the correct answer. This is not true in this case. Okay. Species richness increases with increasing area? Yes, but up to a certain limit? Yes, it has been given in the very first statement. And the last one, the relationship between species richness and area is rectangular hyperbola for a wide variety of taxa. Okay, so this is also the option that has these, all the lines are given in your text. Okay, now moving on to the next question. The gaseous plant growth regulator is used in the plants too. What is the role of that particular gaseous plant growth regulator? when we are discussing different plant growth hormones, we have ethylene, auxin, cytokinin, right, gibberellins, abscisic acid. So we have to learn all the functions, okay, what are the different roles that are being performed by those hormones, and on the respected questions, on the respective questions, you can be given the certain roles and you have to answer. Now here we have been asked about the gaseous plant growth regulator, and that is nothing but ethylene. It is the only hormone that is present in the gaseous form, right, that is present in the gaseous form. So we will understand and learn a few characteristics about ethylene. Ethylene is also named as the fruit ripening hormone, okay. It is also called as the fruit ripening hormone. It is another name for the same. Now, ethylene was basically discovered by H.H. Cousins, okay. You are given such questions in the form of matches, okay. You have been given certain scientists over there. On the other hand, you will be given some hormones and you have to match, okay. So H.H. Cousins is correct for the discovery of ethylene. Now, how did he discover that? He observed that there was a pile of oranges and they were the ripened oranges, okay, pake hue santre, they were there, and along with those there were kept certain unripened bananas, okay, and in the presence of those ripened oranges, those bananas also became ripened, okay. That means something was being released from those ripened oranges, okay, and later on it was discovered that it was the gaseous ethylene that was being emitted from those particular oranges, okay, and that were leading to the ripening of the bananas also. If you observe in your home, you bring guava, okay, it is having such a strong smell, okay, and if you keep guavas with some other fruits, if they are less ripened, they will ripen along with them, okay, and that is only because of the hormone, that is the gaseous hormone ethylene, okay. Now, H.H. Cousins discovered, and if we are asked about the precursor of ethylene, is, okay, it is not working now, I need to erase it all, okay, alright. So we have the what I was discussing, okay, we are talking about ethylene and we have H.H. Cousins who discovered it. Now, if we are talking about the role of ethylene, we have basically ethylene is performing dual function. Okay, what is its dual function? There are plant growth regulators that are the promoters, and there are some that are the inhibitors, okay, that is abscisic acid, that is the inhibitor. If we talk about cytokinin, auxin, gibberellins, they are the plant growth promoters. But here comes our ethylene. It is performing dual functions, okay. So what are the dual functions? It is acting as the inhibitor also as well as a promoter, okay. So what are the dual functions? Let us understand. We have, if we are talking about its promoting functions, okay, its role as a promoter and its role as an inhibitor. So what does it do as a promoter? For example, it is performing the role of stem elongation. So it is a promotory function, right, in the rice, okay. We know when we sow rice, they are the deep water plants because the fields of rice are, they are flooded with the water because it requires that condition, okay. And in that case, the stem has to elongate, otherwise it will drown, okay. So it has to elongate. So the function is being performed by ethylene, stem elongation. It helps to increase the number of female flowers in case of cucumber. And what will happen? See, more the number of female flowers, more is the number of fruits that the particular plant will be producing, and hence more productivity. So it increases the number of female flowers. in case of many plants such as cucumbers. Okay, this is also a promotory function. Next, it helps to break seed dormancy. Okay, we have abscisic acid that is basically promoting this dormancy period of seeds, but this ethylene is acting antagonistic to it. Okay, we have other hormone that is gibberellin that is also antagonistic to particular abscisic acid in case of seed dormancy, and similarly ethylene is doing the same. It is breaking seed dormancy. So it is again a promotory function, breaking seed dormancy. Okay, now what is the inhibitory function? Okay, so we need to understand that if a plant is basically going through some stressful conditions, okay, or we can say the plant requires to shed off its leaves, to shed off its fruits, that is something inhibitory, okay, and that is promoted by ethylene in certain cases. For example, it helps in senescence of leaves, that means aging of leaves. We know in the autumn season all the leaves are moving towards their senescent phase and they are falling, the fall season, right? Senescence of leaves is promoted, and it is, and if they are falling, that is the abscission, okay? So abscission of leaves, and along with that it is performing a very important function, that is thinning of fruits, okay? That is not being done naturally by the plant body. If we are applying ethylene to plants, what happens? See, if there is a tree, it is having a lot of fruits. A lot of fruits are there, okay? They are not ripened yet, they are just the baby fruits, okay, and they are in the raw stage, they are in the baby stage, very young fruits. Now what is happening, this particular plant is not having sufficient amount of nutrients to lead to the ripening of all of those fruits, okay. For the growth and maturity of all of the fruits, it is very difficult for the plant. In that case, what happens, we spray ethylene, okay, not spray ethylene, basically we treat the plant with ethylene. It is a gaseous hormone, because so what will happen, it will lead to the shedding of excess of fruits, okay. In the raw stage only, they will simply shed down, so that the remaining fruits on the tree get the proper nourishment, okay, and they are fulfilled with whatever they are requiring, okay. So basically abscission of leaves and thinning of fruits, the term that we use for this particular process is thinning of fruits, okay. Thinning of fruits doesn't mean the apple is this broad and it will become thin. No, thinning of fruits means shedding of fruits when there are excess of fruits on the plant, okay. So, and not in all, like there is cotton, cherry, such fruits, okay, such fruit trees are treated with ethylene for thinning of fruits. There is the mention of the examples also. I will mention cotton, cherry and walnut, okay. Their thinning of fruits is done. So these are certain promotory and inhibitory functions, that is the dual functions performed by ethylene. Now switching back to the question says, gaseous plant growth regulator is used in the plants to induce seed formation in conifers, no. Promote bolting in cabbages, again no. Promote female flowers in cucumbers, yes, I just discussed this. Increase length of the sugarcane stems, absolutely no. So this is helping in the increase in the length of stem, but in case of rice, okay. So we have rest of the three functions. Can you tell me which particular hormone, only single hormone is performing rest of the three options, rest of the three functions? Can you tell me in the comment box which particular hormone is that? Option 1, option 2 and option 4. These are the functions that are performed by gibberellic acid, or you can simply say gibberellins. Okay, they are responsible for inducing seed formation in conifers or promoting bolting in cabbages. I discussed bolting process in the previous lecture with you. Increase length of sugarcane stems to increase the yield. Very good, Govinda. The correct answer is gibberellin. Okay, yes. Now moving on to the next question. Here we have, in 5 kingdom system of classification, how many kingdoms include the organisms with only photosynthetic mode of nutrition? Can you answer this question? Only photosynthetic mode of nutrition. So I will discuss first of all what are these 5 kingdoms. See, 5 kingdom classification was given by R.H. Whitaker. Okay, so we can simply say the Whitaker classification system. Okay, so R.H. Whitaker divided the organisms or you can simply say classified the organisms into 5 kingdoms. Okay, and what were those kingdoms? First of all, yes, the answer is right. Rohit, Shuvankar, Pragati is saying 2. Okay, let us see. गलत आंसर भी आ रहे हैं कमेंट बॉक्स में. So we need to discuss this concept again, right? So R.H. Whitaker classified the organisms into 5 kingdoms. The first one is Monera. Okay, I will be only discussing the type of nutrition, okay, in these particular member of kingdoms. Okay, so next we have, what I just did, okay. Okay, so Monera. Second one that we have is Protista. Third kingdom, tell me in the comment box everybody, is the fungi. The fourth kingdom that we have is kingdom Plantae, and the fifth kingdom that we have is Animalia. Okay, now speaking of the nutrition, okay, if we are considering the kingdom Monera, now we know the members of Monera can be photosynthetic. They can be photosynthetic, right? They can be chemosynthetic also, right? They can be saprophytic also, right? As well as parasitic. Okay, so all the mode of nutritions are found in the members of the kingdom Monera, right? So that means over here if we see this particular, these two are representing the autotrophic mode, right, and rest of the two are representing the heterotrophic mode of nutrition, right? Now if we consider kingdom Protista, okay, here we have so many organisms. We have photosynthetic protists, we have euglenoids, we have, right, that can perform photosynthesis also, and that can switch to heterotrophic mode of nutrition also. There are certain that are saprophytic like slime molds, okay, and also holozoic. Okay, so here we have both type of nutritions. First of all, they are photosynthetic. If we are talking about their autotrophic type of nutrition, they are only photosynthetic. They are not chemosynthetic, okay? Like monerans, they are not chemosynthetic, they are only photosynthetic, okay? They can be saprophytic, right? They can be parasitic, okay? And at last, they can be holozoic also, like animals, okay? So this is also we have been given. Now comes the third kingdom, that is fungi. What happens in case of fungi? Fungi is the kingdom in which the members are only heterotrophic. Okay, if you are given the question, this particular question has been asked about the only photosynthetic, right? If you are given the question only heterotrophic, the correct answer will be fungi, okay, as well as Animalia, right? They are only heterotrophic. They cannot perform autotrophic mode of nutrition, okay? So if we are talking about fungi, they can be saprophytic, they can be parasitic, Now, the last two that we have kingdom plantae. See, there are certain plants that can perform heterotrophic mode of nutrition also, right? Like we have the pitcher plant, we have the Venus flytrap, right? But in R.H. Whitaker 5 kingdom classification, those particular type of plants were not given any place. Okay, so he said that the kingdom plantae, the members of the kingdom plantae are purely photosynthetic. Okay, so they are purely photosynthetic according to R.H. Whitaker. Okay, so they are photosynthetic. Right, and at last, speaking of the kingdom Animalia to which we belong, we cannot perform photosynthesis. There are so many other animals that cannot perform the autotrophic mode of nutrition. So we can simply mention that these are heterotrophic. Okay, clear with this everyone? So the correct answer is simply the first one. That is, only one kingdom is there according to the 5 kingdom classification given by R.H. Whitaker. That is the kingdom plantae, that is purely photosynthetic. Clear with this? Pragati made mistake in this question. So I hope it is clear with you now. It is not 2, it is only 1. Is it clear with everyone? Can we switch to the next question now? The next question says, genetic code. Again, we have genetic code show genetic codes show degeneracy. It means, so what do we understand by degenerate nature of genetic code? Okay, so I will discuss all the salient features of the genetic code with you, and then we will switch back to this question. Okay, can you please repeat before question? It is asking about the only kingdom that is having the members that are purely photosynthetic. So I have explained it in the form of this table right here. Okay, so these are the different modes of nutrition in the 5 respective kingdoms, and we can see that only kingdom plantae is having the members that are purely photosynthetic according to R.H. Whitaker. Okay, so we have first as the correct answer. Okay. Okay. Now, genetic codes show degeneracy. Okay. Sidesh Borkar and Shuvankar Kundu, they are answering. Please do not spam it. Okay. So, we have the, I will discuss the answer, and yes, you have given the correct answer also, but we need to understand the concept along with it. Okay. So, let us revise all the characteristic features of the genetic code. Okay. So, what are the salient features of genetic code? You already know the answer, that is absolutely perfect, but you should know rest of the features also, and if you know them also, but you have to revise, right? So the first one that we have is the genetic code is a triplet. Okay, I will do this revision part quickly for you. I know you are already smart enough. Okay, Ratan is editing master. Wow. So he is answering 2. Shweta Kumari is also answering 2. Yes, that is absolutely correct. But rest of the features are, first of all, genetic code is a triplet. That means if we are writing a genetic code, we are using 3 alphabets. Okay. We are using 3 alphabets, and those alphabets will be representing the 3 nitrogen bases. Okay, so we have 3 nitrogen bases, okay, that will be represented by the 3 alphabets. Okay, so genetic code is a triplet. The second characteristic feature is our genetic code is comma-less. Okay, that means it is read in a contiguous fashion. If it is written over there, it is read in a contiguous fashion when the translation will be done. Okay, the process of translation will be done when the mRNA will be read to form proteins. It will be in a contiguous fashion. Okay, the third characteristic feature is the one that has been given in your question, that the genetic code is degenerate. Okay, So the genetic code is degenerate. What do we understand by this? See, degeneracy says one amino acid can be coded by more than one codon. Okay, what do we get by this? That for example, I have the codon UUU. Okay, I also have the codon UUC. Okay, both of them are coding for phenylalanine. Right? Both of them are coding for phenylalanine. That means amino acid is just one, that is phenylalanine, but it is being coded by more than one codon, right? This one also and this one also. So this is the degeneracy. Okay. Now the next characteristic feature that has been given over here in your NCERT is your genetic code is basically nearly universal. What do we understand by the universal nature of anything? When we use the term universal, what do we understand? That is applicable to everything in the universe, right? That means it is, if I am talking about the genetic code, if UUU is coding for phenylalanine in my body, it will code for phenylalanine in the body of an elephant also, a dog also, any organism we consider. Okay. मैम एक चीज़ पूछना था, जो अमाइनो एसिड वाला चार्ट याद करना है क्या, उसमें से पूछेंगे क्या? सही, पूरा चार्ट याद करने की आपको ज़रूरत नहीं है, आपको पता होना चाहिए बहुत अच्छे तरीके से कि AUG का क्या फंक्शन है. ओके, इट परफॉर्म्स द डुअल फंक्शन. अभी मैं आपको बताऊंगी आपके स्टॉप कोडॉन्स कौन से हैं. आपको मैंने पीछे जो क्वेश्चन हमने डिस्कस किया उसमें बता दिया, ऑक्रे, एम्बर एंड ओपल. दैट इज़, अब आप मेरे से हिंदी में पूछ रहे हैं तो मैं आपको हिंदी में एक शॉर्टकट बता देती हूँ. ठीक है. सो वी हैव UAA, UAG and UGA. We generally forget this particular sequence or these three codons. See, what I used to, this is a very stupid one I know, but I used to recall it this way only. This is for one of my fellow colleagues also helped me learn this in the same way. He said that I tell in the class, Ullu a, okay, Ullu a, okay. Then second one is Ullu a gaya, okay, that is UAG. And then Ullu gaya, that is UGA. Okay, पहले आपने उल्लू को बुलाया, उल्लू आ गया, and then उल्लू गया. Okay, so this is how this. I know this is a stupid way of learning, but you are learning, right? So UAA, UAG, and UGA. All right. Now we have the nearly universal characteristic. It says that if the codon is coding for some particular amino acid in one organism, it will code for exactly same amino acid in rest of the organisms also, right? But we are using the term nearly. What do we understand by this? That means there are some exceptions also, okay? So here we have the example, let us say UGA, right? UGA is otherwise a stop codon. That means it is not coding for anything. It is stopping the process of translation. But in case of mitochondria, it codes for tryptophan. It codes for tryptophan. Okay, so this is the nearly universal nature. Okay, it is not coding for tryptophan otherwise, it is the stop codon, but it is coding for tryptophan in case of mitochondria. Okay, in the mammalian mitochondria we observe this. Okay, then comes the fifth one, that means start codon. Start codon is AUG. I discussed it with you, and AUG performs dual functions. Thank you so much, Radhe Krishna. We have the function that is dual function. It codes, it is acting as a start codon also, and it also codes for methionine. Okay, so it codes for methionine also. Okay, and it acts as a start codon also. So these are the dual functions that are being performed by our AUG. So these are some of the salient features of the genetic code, right? So the question asks, genetic codes show degeneracy. Now this characteristic feature, it means option 1 says in mRNA codon is read in a contiguous fashion. No, this means the genetic code is comma-less. This is the salient feature, right? Then the second one is amino acids are coded by more than one codon. This is absolutely correct. Third one says one codon codes for only one amino acid. This is the universal nature of the codon. I will write it for you. This is the universal nature. And the fourth one is each codon is independent and one codon does not overlap the next. Yes, this is another characteristic feature, that the genetic code is non-overlapping. Okay, if I am writing a genetic code, let us say we have AUG, UGA, AUC, CAU and so on. Okay. Now, if I want to read it, I cannot overlap the code. Okay. If here is AUG and here is UGA, I cannot overlap it like GUG. You get it? This is a whole sequence. You cannot simply overlap it. You have to read it in the triplets only. Okay. Understood? Alright, very good afternoon El Dorado. You almost missed the class. There is the time for the next slot now. Okay, so the so this is the universal nature, and this characteristic feature is the genetic code is non-overlapping. Okay, so here we have the correct answer for the given question, and I hope the concepts are clear with you. Ma'am, GUG and ambiguous codon. Yes, that is also true. Okay, now while we are talking, there is an exception while we are saying this. Degenerate. Okay, there are some codons that are non-degenerate also. Okay, for example, AUG. AUG is non-degenerate because the methionine amino acid is coded by only AUG. By no other codon it is being coded by, methionine is being coded by only AUG. So this is the over here the degeneracy we saw, but the non-degeneracy is also there, for example in AUG. That is an exception to it. Okay, clear with this? So I hope the concept is clear with you. Please prepare well for your exam, right? Please pass with the flying colors also, so that you get to have a seat in great medical colleges in India. Okay, so the next slot is still there. Please stay tuned. We have the third slot in which the rest of the chapters, the questions from the rest of the chapters will be discussed by our faculty Aman Vishwakarma. And have a nice day everyone. I'll see you in the next session. Bye-bye.